The transcription, I want to stop from where?
Still from altro, and registra e trascrivi.
Done. Okay. So, hello everyone. Today is my pleasure to introduce you Silvie Bernatova. She is a junior researcher here at the IPCF CNR in Messina, working on the project Samotrash. Her research is mainly focused on optical forces and their application on biological molecules or environmental sample, as in the case of this project. Please, Silvie, the stage is yours.
Okay, perfect. Thank you. Ciao a tutti. It's my pleasure to give you a talk. Probably, as you notice, I'm your colleague from this September. And originally, I'm from Institute of Scientific Instrument in Brno, Czech Academy of Sciences. And I would like to briefly introduce my home institution and my way how I get to Messina, and I prepare talk which summarize my research during my PhD, my postdoc, and what I would like to do here, and what is my plan for the future. So, as I said, I did my PhD at the Institute of Scientific Instrument, Czech Academy of Sciences, which this institute is located in Brno, which is pretty close to Vienna. My supervisor was Pavel Zemánek, who is also the head of microphotonics department, and I was in the group of biophotonics in optofluidics. Our main research was focused on application of optical tweezer and Raman spectroscopy to biomolecules, biological objects, and investigation of some living cells like bacteria, yeast cells, and monitor the external stimuli on this object. The history how I get here is connected with Maria Grazia Donato. So thank you, Maria. That in 2016, if I remember well, my supervisor has a collaboration or bilateral project. So I spent here in Messina 1 week. And I started some experiments with cells, and after this, I was here several times with COST project, and in 2021 I applied for project for junior researcher for traveling. So I spent here almost 1 year, and during this period, Pietro Gucciardi said me that there is an opportunity to apply for the position at CNR. So I applied, and I am here. Okay, so the main motto of my talk and what is the take-home message is that the light is not just only for lighting. Light can be used for transport, trapping, sorting, analyzing, characterizing of object, and self-arrangement. So I don't want to go very deeply about optical manipulation because you have director, which is author of very famous book, Optical Tweezers and its application. So it will be just only brief, only slide on the cover what is optical tweezers. So outlook of my talk will be, I will mention some application examples in Raman tweezers for detection of microbiological object. This work was done in Brno, in my home institute, or in my previous, not home, previous. My home is right now here at IPCF. So in my previous institution, and I would like to show you some results which was done during this COST project, during this bilateral project, and during the mobility project for young scientists, and what I did during last 3 months here at IPCF. So what is optical tweezers? It's not necessary to go very deeply. Optical tweezer is a tool how to manipulate with small object by light. Small object means object from the several nanometers to several tens of microns. The most common configuration of optical tweezer use highly focused, very often Gaussian beam. And in the focus of this Gaussian of this beam can be trapped object with the mentioned size and manipulate with them. So optical tweezer regime can be divided in 3 main groups, not just only in biological system, but also in general for the particles which are much smaller than the used wavelength, more or less comparable with the wavelengths or with the focus of the beam, and much larger. In my talk, I will be focused on the particle with the size much smaller than the wavelength and more or less with the size of this wavelength. So it was a brief introduction of optical manipulation techniques or optical tweezer. And sorry. And I would like to show you how it looks like in the lab. What you will see on this video is an amoeba. It is unicellular microorganism, and we trapped inner part of this amoeba. Amoeba has the ability to alter its shape, and you can see that there is no some stress behavior of this microorganism. Probably amoeba doesn't know that that some inner part is manipulated or under investigation. And I think it's pretty nice video how it works in lab. Sorry. So it was optical tweezer. And what is Raman tweezer? Raman tweezer is the combination of Raman spectroscopy and optical tweezers, sometimes call it laser tweezers Raman spectroscopy. And as I mentioned, in the focus of this beam, very often we use microscope objective to trap object. We collect the Raman signal, where is a hidden information about structure of trapped object, structure of matter, I mean. And with evaluation of the spectra, we are able to find some information about the object or follow external stimuli or identify this object. Very, very common is configuration with single beam tweezer. So it means that the same beam is used for trapping and also for exciting of Raman scattering. Or we can use multi-beam Raman tweezers. It's very suitable in the case that that you would like to trap some object with not very symmetrical shape. We can use some diffractive element to split it or divide the beam to several beams, trap some parts, and use another beam like as a Raman probe, and to select which part of the trapped object will be investigated. Suitable wavelength for this analysis is from visible to near infrared and infrared. And all the time it's a little bit playing with the wavelength. And in the next slides, I will speak a little bit deeply about the situation. So this is the main principle, or this is the main principle of Raman tweezers. Here is one of the typical experimental setup. It's not very complicated. You can see that there is few lenses. I used single beam fiber in my setup in Brno just to clean the beam. So it means that the quality of laser wasn't enough. So I used a single beam optical fiber to have a pretty nice Gaussian beam. There is some mirror, some filters, objective which is focused. There is an investigated object, and with the same objective we collect the Raman spectrum, and the Raman light is delivered to detection part of the setup. One of the main positive of Raman spectroscopy is that it's pretty fast method. You are able to see almost online external stimuli or what is in the trap, or identify the object. I spoke about Raman spectroscopy, and I remember at university when I read about first observation or about the name of Raman phenomena, there was written something like, Sir Chandrasekhara Venkata Raman was the first guy who observed the Raman phenomenon, and it sounds like a little bit, he was the first who observed this phenomenon. But to put it a little bit in context and just for fun, and I'm experimental physicist, so I know how complicated is somehow is to observe the data. Keep in mind that in this period, it was 30 years ago, 30 years before invention of lasers. So, for example, Raman used the light from the sun, and he used lenses which has 2 meters in diameter. He hasn't CCD, so he used a photographic desk, for example, or he was able to build his own spectrograph or measure the spectra. So it's some notes about, and what was the biggest surprise for me? It was when you downloaded his famous articles, A New Type of Secondary Radiation. You recognize that there is only 3 paragraphs. And this 3 paragraph looks like as a report to the boss. Okay, I had this, I observed this, and data are available. And this is the base of Nobel Prize for Raman, which was Raman observing Nobel Prize in 1930. So it was just only brief story about the Raman spectroscopy. And you can read that Raman spectroscopy is powerful tool and blah 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 about what is the limitation of Raman tweezer. As I mentioned on the slides of optical tweezers are able to trap particles from few nanometers to several tens of microns. But it is not the same in the case of Raman tweezers. The main limitation of Raman tweezers is that the Raman phenomenon is pretty weak. It's about 14 orders lower than with the fluorescence. So with optical tweezers, you are able to trap super small object in the range of tens of nanometers, but you are not able to observe the Raman signal. I think that below 500 nanometers is pretty challenging how to observe Raman signal. So this is one of the limitation of Raman tweezers. If you would like to identify the trapped object, you have to build a database and the technique how to evaluate, how to connect it, measure its spectra with the spectra which is filling the database. Also, seeking and investigating of object is not so easy, especially in the case of particles which are below the diffraction limit. And with optical trapping, if you trap some larger object, it is not easy to manage the orientation of the object in the trap. This is the reason why I think was introduced some holographic tweezers or some multi-beam tweezers to be able to rotate or manipulate with the object. So as I said, I would like to introduce experiment which was done in Brno during my PhD, because the title of my thesis was something like application of optical manipulation and Raman spectroscopy to microbiological object. And I would like to show you some results, as I said. The first is, what is the motivation to trap bacteria or measure bacteria? It is connected with clinical practice. Nowadays, practice is when you go to the medicine to the doctor, I mean medical doctor, and you are under attack of some infection. Very often, the doctor just only gives you some broad spectral antibiotic without any investigation of the infection. It's because that investigation of infection is pretty time consuming. Because if the doctor collects some sample, for example, from your neck, eye, ear, or urine, blood, it takes at least 2 days to cultivate this bacterial culture. If you would like to test it on antibiotics, it takes 1 more day. And in the case of bacteremia, bloodstream infection, or even sepsis, there is no time to wait so long, because in the case of sepsis, you can be dead in the range of hours. And we believe that the Raman spectroscopy can be solution, because in the real time we are able to identify the object in the range of hours. We are able to say if the trapped bacteria is sensitive to antibiotics, and another possible application is to follow the external stimuli. As I said, the first step is identification of bacteria by Raman tweezers. So it means that we build the spectral database, we trapped some bacteria, we collect it, and with this tool we are able to distinguish which kind of bacteria is in the trap in the case that the database is filled with this spectra. So the summarize of this slide means that we are able to skip the cultivation step, because for Raman tweezer analysis, it is necessary to have some hundreds of bacteria, and hundreds of bacteria can be found in the sample which is obtained from your neck, for example. So if you identify, or if someone identify the bacteria, the next step can be how these bacteria are sensitive to antibiotics. So the next experiment which I would like to show you is experiment when we monitor the cultivation process in the presence of antibiotic in microfluidic chip. This is the design. This is the design of microfluidic chip. We injected cells in the presence of antibiotic. This is the scheme of experiment, and the main idea of the experiment was that in the Raman spectra, we know that some bands are connected with DNA and RNA, and we suppose that in the case of sensitive bacteria, I mean sensitive to antibiotic or resistant to antibiotic, we can follow this response, and we can distinguish which bacteria is sensitive and which is not. This is the result. Here you can see the Raman spectra of bacteria which are sensitive and cultivated in the presence of antibiotics. It is clearly visible that the response of DNA and RNA decreasing in the time. There is a time evolution of the Raman spectra, and you can see that after 5 or 6 hours, the response of DNAs in the case, the red one is the bacteria which are resistant to antibiotic, the blue one is which are sensitive to antibiotics, and you can see that in the range of 5 hours we are able to distinguish or to set which kind of antibiotics are sensitive to antibiotics and which are not sensitive to antibiotics. So it means what you see here is the response of DNA. In the case of resistant bacteria, you see that there is some fluctuation of the signal, but after 5 hours, the bacteria are able to survive, are able to divide, and the response of DNA came back to the initial value of the experiment. And what is one of our masterpiece is that if the doctor collect some samples, it is not in pure water. So you have some surrounding media, and this surrounding media can have a huge response in the Raman spectroscopy. So we developed the technique which call it spectral mixture analysis, how to divide the response of surrounding media if you collect also the spectra of surrounding media from the response of the cell, and it's all the time same story. If you are able to distinguish the response of the surrounding media and the cells, you are able to connect it with database, and you are able to identify the trapped object. And okay, so with Raman tweezer, we proved that we are able to identify bacteria. We are able to say if this bacteria is sensitive to antibiotics. So, and what will be in the situation that you know that you are under attack of this kind of bacteria and it's antibiotic resistant. We use this kind of experiment, which where we have a bacteria which were resistant to antibiotic, and we infected this bacteria by viruses, by bacteriophages. Bacteriophages attacked adversely just only one kind of bacteria, and also with Raman tweezers, we trapped this bacteria and we follow this response, and we was super super surprised that after 5 minutes we are able to say how these bacteria react on external stimuli in the form of bacteriophages. In 5 minutes it was completely clear if these bacteriophages are able to attack this bacteria. So it's not just only about to identify or to collect the spectra from bacteria. Bacteria also can serve as a sensor. You can have a sensor in the form of bacteria. It means that you know exactly the response of this bacteria and you can follow the changes. So it can be used for example, external stimuli in the form of antibiotics, bacteriophages, or for example, temperature, some disinfection. So it is my result from the PhD studies and from the postdoc. And right now, let's move to the detection of microplastics. In 2018, European Chemical Agency reveals that the microplastics is one of the hot topics. Completely, it's about 15 hot topics, and probably it's not necessary to do some introduction or motivation why it's necessary to measure microplastic. Microplastic is an object, is a plastic object with the size from 1 microns to 5 millimeters. Nanoplastics are plastic object below 1 microns. And how to measure this microplastic, because there is a huge technological gap, because most of the common techniques fails in the case that you would like to measure some environmental samples, because these samples are heavy contaminated, it's full of mess. And as I mentioned before, the ideal candidate for Raman tweezers is the object with the dimension from 1 microns to 20 microns. So also we believe that Raman tweezer is ideal candidate for this analysis. And it's much easier than in the case of bacteria, because bacteria strains, there are hundreds of bacteria strains. So to build some powerful database, it takes lots of hours and hours in the lab. But in the case of plastic, it's easier, because how microplastic grow. It's the degradation of the larger object. So there is only 6 or 7 group of microplastics. So to build a database is not so complicated like in the case of antibiotic, like in the case of bacteria. And also we can apply how to distinguish Raman response of surrounding media, for example. So do you know this picture? I would like to mention these articles, which was published by Nanosoft Lab Group in 2019, and honestly, it was the first article when someone trapped and analyzed the environmental samples. So it was the first one, and the next work continue. For example, in 2021, the group around Sheila McCormick published very similar articles. So it's all the time the same story: trap the object, analyze, and somehow connect it with the database. And also one PhD student from Brno was here for priority cost project, when he trapped and analyzed the microplastic object. The source of the microplastic object was degraded surgical mask. And it is not just only that you are able to analyze, you are able also with Raman tweezer to follow the degradation process. Or surgical mask is a composite material. You are able to determine which layer degrade how fast, for example. So the Martin trapped tens of particles from the surgical mask, and he was also able to see that the layer which is covered by dye who also serve as a protectant, degrade much slower than in the case of some inner layer which degrade pretty fast. So this sample was degraded by UV light for 1 year, and you can see that only in 1 year where this surgical mask was illuminated by UV can be degraded to microscales object. Okay, so it was how it is in the situation of the particles from 1 micron to 20 microns or to several tens of microns, but how it will be in the case of nanoplastics. Because as I mentioned, Raman tweezers are limited for detection of nanoplastics mainly due to two reasons. Low sample volume, so it's mean low concentration, and seeking of object when we are below diffraction limit. Possible solution can be some enhanced Raman spectroscopy technique. So one of this technique was introduced by Barbara Fazio in 2016, when she used optical pushing for create of plasmonics aggregates, and this plasmonic aggregate means that you create aggregate which consists of plasmonic particles. Here is the temporal evolution of generation of this aggregate. You can see that at the beginning there is nothing. After 70 minutes there is a huge aggregate which can have several microns, consist of these small particles in the range of tens of nanometers. There is lots of hotspot, so the possible application is to excite some surface enhanced Raman spectroscopy. So I continue with this work. I implemented two microfluidic chip, and I observed pretty nice enhanced effect on some protein. And in the case that we used the wavelength which was far away from the main plasmon resonance, there were no source effect. Okay, so after this, I said, okay, can we use the same for microplastics? Because, you know, in the case of protein, there is a small molecules. But the plastic, even if we speak about nanoplastic, it's a huge object in compare with molecules. So I mix it, my plasmonic particles with small plastic particles. I prepare some aggregate, and I observe some signal of plastic. But the enhancement effect was not pretty high, because in the case of real SERS, surface enhanced, I mean this kind of enhancement of Raman spectroscopy, it's the enhanced factor starts at 10 to 3, and I observe pretty small enhancement effect in compare with what is the real SERS. I said, okay, it's a little bit strange how it is possible. So I changed the wavelength, and I revealed that in the case that I am far away from the plasmon resonance, I observe very similar effect. And I think, okay, how it is possible? What is the main feature of some metallic structure? And I reveal that it is not the effect which is connected with localized plasmon or surface plasmon polariton. It is the effect which is connected with temperature gradient. So the main idea of this experiment is that we use optical forces to push the particles to create an aggregate, and also we create a temperature gradient which attract the plastic particles. So locally you increase the concentration of particles that you are able to observe Raman signal. So we break the detection limit of Raman spectroscopy, not due to the plasmon resonance, but due to the higher concentration of the particles. But how it will be with optical pushing in the case that we will use different wavelength and far away from the plasmon resonance, because at the beginning I spoke about the cells, so it is necessary to use the wavelength which is close to the main plasmon resonance. And Maria Grazia prepare us calculation of optical forces and different orientation of nanorods, and the result of the calculation is that for any wavelength and any configuration, all the time the particles will be pushed. So it means that with the wavelength which we used in our experiment, we are able to create an aggregate. So it means that we are able to create also the temperature gradient. And this is the result when we do this experiment with green wavelength, red wavelength, and near infrared wavelength. And this is the temporal evolution of the peak, which we can label it as a marker, if there is a marker of polystyrene, if we are able to attract enough particles to see Raman response of the polystyrene. And this is our masterpiece. We are able to detect it 27 nanometers particles. Here you can see the Raman spectra. The yellow one is the bulk polystyrene. This is the spectra after 1000 seconds of aggregation, and this is the spectra at the beginning. So it means this is the background. Okay, and it was what was done during my mobility project here in Messina, and also in my institute in Brno. And what I would like to do here is improve the microfluidic and implemented whole procedure to microfluidic. What is microfluidic? It is a system that manipulates a small amount of fluid using small channel with size from 10 to 100 micrometers. And why we would like to implement the process to the microfluidic? It's because it's a easier to control environment, and you can totally control the environment. You can easily automatize the measurements, and for some, how to say, external user, they are not able to have setups 2 meters per 1 meters on optical tables and prepare the samples with the spacer. So it's much easier with microfluidic. You just only injected the samples, and it will be work like automatical way. And I prepare video of some guy from my previous group. He designed some microfluidic platform. It's half in Czech, there is in English. So he generates some droplets inside of microfluidic, and I would like to show you what is everything possible to do with microfluidic. There is a testing of droplet with laser, so he is able to count every droplet. He is able to change the size with electrophoresis to sort the object. And here is some bacteria which were trapped in the droplet. And there are different shape of the droplets. So it was just like commerce. So I would like to implement it here, some microfluidic device. No. So last 3 months I worked with this guy. His name is Iker Gomez from San Sebastian, and here you can see our lab. There is Raman tweezer. This is a syringe pump. This is what we see on the screen, and I prepare nice video. It was from the beginning. You can see there is a microfluidic channel. There is a flow, 5 microns object, which was trapped, right now escape. And there come a new one. microscope, syringe pump. And the next video is about testing of the highest velocity. If we are able, with pretty high velocity, if we are able to trap object, and what is the limit. You can see that the particles are pretty fast. Sometimes they skip. So it's all, and I'm looking forward that I will collaborate with you, and I will meet you on some department meeting. Thank you for your attention, and may the forces, optical forces, be with you.
Thank you very much, Silvie.
Is there someone, or do you sleep?
Yeah, sure. Let me stop the registration.
Okay.
Okay.